I had the pleasure of presenting at EHA on the subject of Pirtobrutinib resistance. Now, as you know, many patients do develop progression on covalent BTK inhibitors, which include ibrutinib, acalabrutinib, and zanubrutinib. And when they do so, they often acquire mutations in the C481 residue of BTK. Non-covalent inhibitors like Pirtobrutinib are showing significant clinical activity in the setting of these pre-existing mutations. There have been a couple small reports of mutations at other sites in BTK on Pirtobrutinib, and we wanted to investigate it further in the Bruin population. Now, the Bruin population is heavily pretreated CLL patients who received Pirtobrutinib on the phase 1 study, and at this point we have 49 patients who have both baseline and progression samples that we could evaluate by targeted next-generation sequencing of 74 genes. And then for patients who had acquired mutations in BTK, we also performed manual review to see if they were present at baseline. So what we found was that at baseline, among these 49 patients, almost all had had disease progression on a prior covalent inhibitor, and in almost all cases that was ibrutinib. About half the patients had pre-existing mutations in BTK or TP53, and there were 10% of patients that had PLC gamma 2 mutations. All the patients, regardless of these mutations, had a similar response rate to Pirtobrutinib as patients without mutations, which was good. If we looked at the acquired mutations, 71% of patients acquired new mutations, and 55% of these patients acquired mutations in BTK. 16% acquired mutations in other genes, and 29% had no acquired mutations, suggesting a different mechanism of resistance. And then what's very interesting is if we look at the acquired BTK mutations, we actually see that the C481 mutations associated with covalent resistance almost always disappear. And what comes up instead are the T474 gatekeeper mutation and the L528W kinase impaired mutation. We also found that about a quarter of those mutations were pre-existing at baseline at very low levels, suggesting they were acquired on the prior covalent inhibitor. So these data, I think, are very important for identifying the primary mechanisms of resistance to Pirtobrutinib in the relapse refractory setting, and also identifying that many of these mutations do come up during the prior covalent inhibitor. We need more work to understand which mutations occur most commonly with which covalent inhibitor, and also whether they have any impact on Pirtobrutinib response when they're present at a low level. With current data, these data suggesting that the low level does not preclude Pirtobrutinib response. We are having a further evaluation of a more expanded cohort, as well as of other genes that we saw with acquired mutations at time of resistance.